who is an investigator at the Lunenfeld-Tanenbaum Research Institute, and he's also an assistant professor at the Department of Molecular Genetics and Statistical Sciences here at the University of Toronto. So among many other things, he works in machine learning with biomedical data, looking at single cell and cancer genomics. So needless to say, incredibly important work. Dr. Campbell, thank you so much for joining us.
Hey, thanks so much for the invitation. Let me reshare this and hit play. And please tell me if that doesn't look okay. So thanks so much for the invitation to speak today. And I think as Radu said last week, it's really nice to sort of give more of a perspective talk than a technical talk that we seem to give so often. And if anyone has any questions, please feel free to just unmute and jump in rather than go into the chat if you like. That's totally okay. So I'm going to give my thoughts on what is a very loosely defined field of data science in biomedical research and whatever that means. The first thing I'll say before starting, normally I start with the disclosure slide that is like trying to convince you I'm not pocketing millions from pharma. I think today really need a disclaimer slide instead that, this is my opinion and one person's opinion is a poor proxy for the truth. So please take it with a pinch of salt, but it's sort of from experience in the world of biomedicine. So I'm going to divvy up today's talk into two different parts. The first is really a sort of introduction to where, whatever you want to call it, AI or ML or data science fits into the analysis of molecular and biomedical data. So I'll briefly go through like what sort of data types do we typically encounter in these settings, and what sort of questions do we actually have about them that we want to answer with these tools, and give you a mini case study on the area that we work in. And then in part two is something that's sort of been on my mind for a while. That's really called Pick Your Problems Carefully, and this is all about are the problems and questions we're going after with our methods really necessary, and are they the best ones that we could be spending our time on? So just to give you a sort of overview of where are we with AI and machine learning in this arena. So this is a figure or a landscape figure from 2020 that just shows you all the different startup companies that exist in the domain somehow links AI with something in health, so drug discovery or some sort of biomarker development. And you can see there's a lot of them, right? This is 240 different startups that somehow associate some sort of data science with health research. So the point I'm trying to get across here is there's a huge amount of enthusiasm and also money lying behind the convergence of these two fields at this moment in time. So it's an exciting time to be in this game. It's also interesting to ask, like, why now? Why is this happening? And I think the answer isn't actually as obvious as some people might think. So you can look at, this is the top one accuracy of various machine learning models and image classification. So you're probably familiar with this huge proliferation of deep learning and image classification. So this is really going along the years from just for 2012 to the present, looking at the accuracy of ImageNet, which, if you're not aware, is this big data set about classifying images, essentially. And you can see from I actually don't know how my mouse works, but you can see from just for 2012 to 2013, there's this huge jump in accuracy of about 15%, which is really unheard of in what was a sort of semi-stagnating field in how well we could classify images. And that was really the sort of deep learning revolution that sparked what kind of followed. And from there was like really impressive gains. They were sort of somewhat leveling off, although they have to level off obviously as you get near 100%. But the point is, we suddenly, since around 2012, have these tools at our disposal that can actually do sort of inference and prediction on these really large data sets. So you can take the same time scale and ask, what's happening in sort of biomedical arena at the same time. So what this graph shows here is it's the number of terabytes of sequencing data. So this is things like DNA and RNA sequencing that we'll come back to later. So it's the number of terabytes of this data that have been deposited in what's called the Sequence Read Archive. It's basically a big online archive where you store all your sequencing data. And what you see is a sort of same thing, right? That around 2012, the amount of data deposited here actually starts to take off. And I think what's kind of fascinating about this is I'm trying to convince myself that this isn't a coincidence, but really the two are sort of disconnected, and actually that just around the time where we have these tools that allow you to do inference and prediction on large amounts of data, we just so happen to have that amount of data coming through in the biomedical domain. And this is echoed across different data modalities as well. It's not just sequencing, it's imaging and others too. So it's kind of exciting to be sitting at this stage where you have the tools and the data that just so happen to have come together. So we can start by talking about that and saying, well, what sort of data do we actually have that we can go after in this big and sort of poorly defined name of biomedicine that I've come up with? So the first thing we spend a lot of time looking at is DNA sequencing data. So I come mainly from a cancer context, so we're often interested in looking at the tumor and asking either what mutations that tumor has that are specific to the tumor, or perhaps the person as a whole. You get sort of mutations in the whole body that are kind of interesting. You know, this also is more topical at the moment because it's the same technology that's telling you the mutations that are in the latest strain of your virus of the moment, which I think is on all our minds. So it's all the same data here. We're also interested in what's known as RNA and protein expression. And without giving a sort of 101 of molecular biology, what this tells you is actually the behavior. It's like a proxy of the behavior of a tumor or a tissue or something similar. So it's going to tell you how is this tumor behaving? Is it sort of proliferating very quickly? Is it growing? Is it dying, etc.? So it's a sort of proxy of behavior. We get epigenetic data as well. So these tell you kind of what regulates expression without really going into it. It's sort of semi-heritable things that regulate expression. Beyond this is a huge amount of imaging data we're also interested in. So you'll know a bunch of these. There's X-ray, MRIs, pathology, images of various types, and these often tell you, what's in the sample. Is there a tumor here, etc. Beyond this, we get a whole bunch of clinical data as well. So there's tumor stage, things like patient, both demographics, but also prognoses. A big one here is response. So maybe you've measured a bunch of patients and you know who responds to a drug and who doesn't, and then I want to go back and start asking questions about what correlates with this and maybe make some hypotheses. And there's so many more that you could come across here. But these are the main sort of data types that exist in our world anyway. So that's the data. What sort of questions do we have about this that we kind of want to use data science, machine learning, statistics tools to answer? And I've kind of divided these up into three broad categories. And they're about really asking what's in a sample, what's different between samples, and what's causing what in a sample. And I'll go through these in turn and give you the sort of spectrum of very simple data science approaches to answering them and all the way to the very complex. So the first sort of one is what's in a sample. So you measure something and you want to know what's in here. So the sort of example you could have is, what mutations are in a patient's tumor, or equivalently, what new mutations are in a new viral strain would be the one of today. And so what you can do is basically just count the number of mutations you find. So in these assays, you basically measure the DNA over and over, and you get what's known as reads. And so this schematic here is that each row is a read, and you can see in my normal, it all looks normal, which is the gray. And then in the sort of tumor sample, you can see this mutation, this green and red is basically the mutation that you would see. And so you can ask how you'd identify this. And well, the simple answer you give is you just count, right? I would just count what's in A, what's in B. And if there's non-normal ones, I'd assign it to tumor. And I think this sort of echoes the history of molecular biology, which is often we just looked at stuff, right? You just looked at a microscope and seen what's there. And it's actually a good approach in many situations, right? But of course, it doesn't work always. And what's fascinating is the same process of looking, is there a mutation? There is, of course, a deep learning solution for it. And in a way, it's actually necessary. It's almost always not as clear cut as this example I've given you. And what's sort of fascinating about this is the raw data you get is actually strings, right? You get strings of A, C, T, and G coming off the sequencer that are the raw data. But what this Google solution does, so Google have this method called deep learning for doing this. They actually render it to an image that you see. They render it to how the human sees it, and then pass that through a deep learning framework to work out whether there's a mutation there. But the point is, I'm trying to get across is for every question we have, there's often a really simple solution and there's often a really complex solution. And the point I'd like to make, we'll come back to later, is even the simplest of solutions has a false discovery and a true positive rate that you can get, right? And so there's always a spectrum of how complex you can go. So that's the first question, what's in this sample? The second question you might ask is, what's different between two conditions? So we talked about the expression of genes, and we'll come back to this later, actually. So this is a proxy for behavior. So I might want to ask, is there more of this gene product in condition A than condition B? Is there more in disease than control? Is there more in a patient who responds to a therapy than doesn't respond to a therapy, right? And you can say, well, what data science approaches am I going to use to answer this question? And the simplest, how do I compare differences in two means? Well, I'm going to use a t-test, right? So the simple thing here is you just hammer at the t-test, and we'll come back to t-tests later because they're really good. So nobody wants a picture of a t-test. So I've got some Guinness here because Gossett was working for Guinness when he came up with the t-test. Of course, it can get a lot more complex than this. So there's some really nice work in this field doing variance shrinkage estimation for these estimators to try and increase the power of your testing. So there's a lot of work in RNA sequencing to do this. So there's a nice example on the right and everything in between. But again, the point is there's this full spectrum of approaches we can take to these problems, starting from the simple and going to the complicated, and they're all associated with some power and some false discovery rate that's worth thinking about. The final one that comes up is what regulates or causes what. So we're sort of touching on something causal here. If you look at causal inference, you meant to identify all your confounders, etc., and you're never going to do that in any realistic biological situation. But quite often we make causal assumptions. So one example is, does a mutation in my DNA cause a change in gene expression? Right, this is a classic question in sort of modern molecular biology. Again, what's the simple thing you can do here? You can use a regression model, right? I can just fit a simple regression model. Again, at the other end of this, well, I was kind of lazy here and just Google deep learning for the problem because you can always find something. But the point is there's a spectrum for doing these problems. So in there's this actually really very nice paper that comes up with a deep learning framework to predict various different gene expression properties based on sequence. They're not exactly equivalent problems, but the point I'm really trying to get at here is there's a whole spectrum for every question we ask about biomedical data, about how complex we want to go with our model space. And it's kind of interesting to think, like, what sort of questions are we asking of this data, right? Is there something in common with all of these? One feature is they're almost always about understanding or some sort of structured discovery or aligning with some existing knowledge about molecular biology. It's very rare we have a pure predictive model where you're going to come along with some supervised machine learning model and ram it into production. They're often not very well defined, which is a strange thing to say, but often we're saying, what sort of cell types are in my data? We'll come back to this one in a second. And the question's actually kind of underdefined, which is exciting because there's a lot of opportunity, but it's also kind of scary. Which brings me to the next point, is that we so often lack a ground truth in our models. And I think we lack a ground truth in two different ways. You quite often don't have the ground truth because it's very hard to measure, or sometimes it doesn't exist. So in the non-existent case, again, if I'm coming up with some cluster model, say I want to cluster biological data, well, what is my ground truth there, right? I'm trying to find some interesting structure in the data, but how can I compare this against other algorithms or approaches? You very often resort to sort of smart heuristics that are kind of domain aware. And I would say actually a lot of our work is about trying to design smart heuristics to benchmark and quantify how well our algorithms work. The other side here is you actually don't have the ground truth data, and it's very easy to end up in a catch-22 here, because oftentimes you design a method where you want to predict or you want to quantify something about your data, right? And in order to show that it works, you would need to have the ground truth data to show that you can recover the ground truth, right? But the catch-22 is often if you had the ground truth, well, you wouldn't need the method you're about to develop in the first place because you just measure that instead. And so there's actually quite a limited scope of problems where you might have the ground truth in some cases, but you want your model to expand to a much larger range. But it's all these sort of things that go around in your head all the time when you work in this domain that you really have to almost have a sixth sense of thinking, what's a smart heuristic I can go after here that's gonna tell me whether what I'm doing is actually working or not. That's the sort of thing that keeps us up at night. The last thing is that actually, you know, we talk a lot about big data and small data and everything in between, and we have the entire spectrum of it here. And what's fascinating is that you can actually have big and small data simultaneously, which limits the questions you can ask simultaneously. And I'll give an example of this in the next slide. Does anyone have any questions at this point, by the way? I'm happy to sort of first mini break if anyone wants to ask anything. But if not, I'll continue. Cool. So I'm just going to give a very brief case study, which is actually what we work on, because I talked in very general terms so far. I want to be a bit concrete about some of this. And this is in this idea of the tumor microenvironment. So I'll give a 30-second biology lesson here, which is when you have a tumor. So this schematic on the left-hand side is interested in the tumor, and the tumor cells, cancer cells, are the very round cells. Very rarely do you ever actually get a tumor that's just a tumor on its own. You almost always have this sort of mix of different cell types that interact with the tumor in very complex ways. So when you resect a tumor during surgery, you'll almost always get cells of the immune system that have infiltrated. You'll get these cells called fibroblasts that are structural cells that hold it together. You'll get growth of blood vessels that grow to support the tumor and tumor formation, right? So you get this whole mix of cells along with the tumor when you do it, right? And these are super important for maintaining and establishing and helping the tumor grow, actually. So there's a lot of research into how we target these cell types with different therapeutics, because you get very complex interactions between them. So it's interesting to think, well, how do I measure this? How do I measure any of this, right? This is a hard thing to measure fundamentally. And one technology we've come up with is called highly multiplexed imaging. And what highly multiplexed imaging does is it basically takes many photographs of a tumor, except each photograph, rather than scattering light, it quantifies the expression of a protein. So each of these images here is exactly the same tumor. It's actually a breast cancer tumor. You can see the sort of the holes in it are actually the ducts of the breast, and the tumors typically form around these ducts. And each of these is a protein, and I can measure the expression. So the brighter the spot, the brighter the expression of the protein in that spot. So what I essentially get is high dimensional images. So rather than an image being RGB in 3 dimensions, I get 30 or 40 of them, where each dimension is the expression of a given protein. And each of these proteins has different functions and tells me different things about the cells and what cells are there. So it basically gives me a very high dimensional picture of a tumor that I can then tease apart and start to work out things about. Okay, it's a really fascinating technology that we have set up in Toronto in various different places. So you might ask, okay, well, how big is your data? How big is it's the sort of question you want to ask, both of you trying to show off to someone, but also because you want to understand what questions you can actually ask about your data, right? So here we measure about 40 proteins, right? We measure, this current technology allows you to measure a subset. There's tens of thousands of proteins. You measure 40 of them, you've got to choose carefully. But 40 is not a very big number, right? So it sort of limits the questions you can really tell about the protein space, right? So I can't really do that much interesting thing about asking which protein regulates which other ones. But we measure this across a cohort of about 500 patients. So I'm going to image about 500 patients worth of tumors, right? So 500 is quite a nice number. I can start to ask some pretty interesting associative questions by the time we get to 500, right? I can ask things about patient survival. I can think about subtyping them. I can maybe even think of some very basic sort of predictive models. What's next? Well, I measure about 1 million cells in one of these, right? So 1 million is a really large number. Now I can start thinking of reaching for my deep learning toolbox if I cared about that sort of thing, and learning sort of rich cellular representations. And at the pixel level, you measure like 100 million pixels, right? So if you wanted to make a statement about pixels in these images, then you're really onto something. But the point is, for a typical cohort we establish, I can look at it in very different ways, get very many different numbers for this N, and that really limits or expands the sort of questions I can ask or answer about about my data. So just to give you a very, very brief sort of insight into one of these questions, right? The question we ask quite a lot is what cell types are in my data. So I have these 100 million pixels, and I want to sort of align it with prior knowledge of the cells that act in cancers, because you're not the first person ever to have done any cancer research, so there must be a way to link this with what we know already, right? So you can ask what cells exist in a tumor, and the answer is we almost get tumor cells. So the cell here is a really nice image. It's much higher resolution than IMC, by the way. It's much smaller. It's like 4 cells. So this sort of blob in the center is a tumor cell. I'll tell you what the CK positive means in a second. And surrounding it are 3 T cells. And you might have heard lots about T cells from recent COVID things, but the fascinating thing about T cells is they also attack cancer cells. So in a set of cases, a T cell will actually go after and attack a tumor cell, depending on various properties of the tumor cell. And that's great, right? Because your body actually has this built-in mechanism to attack tumor cells. So how does a T cell attack a tumor cell? Well, there's this sort of protrusion comes out of the T cell that's part of what's known as the CD3 receptor complex. It actually doesn't matter that much. And this basically latches onto the tumor cell. And if it recognizes it's a tumor cell, it kills the tumor cell, which is good for everyone involved, right? But the point is, this is how molecular biologists think about this data, right? They think, oh, a T cell expresses this protein called CD3, and it turns out that a tumor cell expresses a protein, it's called cytokeratin, but it has its own markers, right? So on the one hand, I have these 100 million pixel high dimensional images. On the other hand, I have this molecular biology knowledge that T cells express CD3 and kill tumor cells that express cytokeratin, right? And so there's a really interesting problem of how do I marry these two very, very disparate fields together, essentially. And so my overall research approach to this that we've been thinking about for quite a while is, can you squish how humans think about biology into a simple statistical model and use that for inference, right? And I'm not really going to go into this, but this was this overall sort of idea I was kind of chase after when I started a research group is, can you somehow replicate how humans think about the data as a statistical model? And then if you do inference, you're going to learn all sorts of different things. So, what does a human think? Well, I think a cell type, like a cell that has a particular function, is defined in terms of the overexpression of a certain sort of marker genes. So for example, from the previous slide, T cells express CD3. And if you think hard enough, and the math here really does not matter, I can actually write down a conditional expectation of a model that replicates this in a way, right? It actually says, oh, if you tell me what the markers of the cell types are, I can run this into conditional expectation, and you slap a noise model on this and some priors, and you can do inference, and suddenly you can actually align this molecular biology knowledge with these like 100 million pixel images of tumors. And so the point is, it's a really exciting time that we have all this new data modalities that are really, really huge, but sort of aligning it with what we know already about the biology is a real challenge, and a really interesting challenge as well. All right, that is hopefully a whirlwind overview of some of the problems we have in molecular biology and some of the ways we're tackling them. I don't know if there's any questions at this point. Please feel free to ask any time, and if not, I'll jump into the next section. So this part is really something I've wanted to discuss for a long time, and I think it's sort of being thought about by the field more generally. And the overall idea is pick your problems carefully when you enter this field of data science in biology, and I think this applies to many, many other areas of data science too. So it's summed up really nicely by this in this tweet from Christine Lum, who's a statistician over in the US. And she says, has anyone done a study on the median number of citations a stats or ML methods paper gets from papers that actually use, and the emphasis here being on the method is actually used and that the paper introduces. And she sort of says she'd be shocked if it's the median number is more than 0. In other words, we're creating a lot of methods in these fields, in the machine learning and to, I think, a lesser extent in the statistics field. And we're building this huge body of knowledge, but is anyone actually using the methods we're creating, or is it just an academic and academic exercise. And so we can look at this in my own field. So I spent a lot of time in working on data analysis methods for a field called single-cell RNA sequencing. So very briefly, the RNA sequencing tells you the behavior of a cell. In the last 10 years, technologies come along to do this in single cells, hence single-cell RNA sequencing. And we're interested in computational methods for it, right? So the first thing you can ask is, well, how many studies are there or data sets? So here you can think of a data set and a study as interchangeable. How many data sets are there that introduce a single cell RNA sequencing data set? So again, there's this time period plotted here from around 2015 to present day. And you can see there's been a remarkable number of growth in this technology, right? So in 2016, there's probably about 100 single cell RNA-seq data sets. Now there's about 1,500, right? So you can ask, well, okay, we've got about 1,500 data sets. How many associated tools or methods do we expect to exist to analyze this data, right? If I was doing this live, I would poll you and see what your expectations were. But it's interesting. It's how many tools or methods that generally analyze or maybe more sort of do machine learning on these data sets do we expect? And you can plot the equivalent graph. And the answer is, if you look at the same time period from around 2016 to present day, there's just over 1,000 methods for analyzing this data, right? And this is kind of astonishing, right? I have roughly 2 computational tools for every 3 studies that actually have the data. Okay, so the analogy for this is each of these data sets is a bit like a photograph album. I wouldn't say it's a photograph, but it's like a photograph album, right? So it's like there's 1,500 photograph albums in existence. And for those, there are over 1,000 software tools that either, I don't know, maybe color correct them or do some prediction from the photographs, right? It's 2 to 3 tools to study. Like, this is completely astonishing. And I think people are starting to recognize that there are a lot of tools we have. And it's kind of fascinating because we have this narrative that we really lack quantitative capacity as a society for dealing with this. And I think this somewhat suggests that we're placing the capacity we do have in the wrong place. So, okay, you might say, well, that's fair enough, but are these 1,000 tools like super unique and necessary, and I'll bring something to the question. And so we can come back to this question of differential expression analysis, right? So this is, again, the question of, is there more gene in condition A than condition B? That's basically what we're chasing here. And except this time we're looking at it at the single cell level, right? So is there more of a gene in one set of cells than another? Again, comparing like health to disease, or those who respond and those who don't. So there's this really nice paper from Luke's Appiah where they basically look at the landscape of tools that are out there. And they found 117 tools for single cell differential expression analysis, right? So this is, it's about 11% of total tools that do this. And so that's a lot. If you imagine, like, each of these is probably like at least a chapter of someone's PhD thesis, right? It's like 117 thesis chapters chasing this problem. So, okay, an interesting question is, do they work well, right? What's being added to this picture? So this really nice paper from Charlotte Sorensen and Mark Robinson in 2018, where they basically benchmarked multiple single cell differential expression methods. So not all of them, but multiple ones. So the table here on the right is from their paper. And so each row here is a method, and each column is basically something they've decided to benchmark on, right? So things like true positive rate, false discovery rate. They also look at things like scalability, so can it handle a large number of cells? They look at speed, is it fast, fast enough to complete? Failure rate, does the software fail? And they marked each of these methods as good, intermediate, and poor. And this is ordered sort of roughly better at the top and worse going down the way. And what you find is, and nobody had really looked at this before, if you use a t-test or a Wilcoxon test, they come out really close to the top, right? And I actually take slight issue with this, because they said a t-test can't handle a complex design, but if you use a linear model instead, you'd be doing pretty well, right? So nobody really thought to, actually just check the baselines here for this simple one. There's actually historical reasons for this. So the field's not completely to blame. When you had very few samples, these very complex methods that did this very fancy variance shrinkage works much better than a t-test. It just so happens when you start measuring hundreds of cells, your variance estimates are very tight and you don't need any of this anymore, or at least not as much. Okay, I don't want anything I'm bashing other people, so I'm going to bash myself for a bit now and talk about the pseudotime estimation problem. And this is what I spent most of my graduate studies looking at. And we're gonna tell the same story. So it's actually a really cool problem. So I'll just talk you through it very quickly. So often cells undergo some physical time process, right? And what I mean by this is they change over time. So a cell will differentiate from a stem cell to another cell type over time, or it might die through a process called apoptosis. But the point is they change over time. But in the current technology to measure this, it's exceptionally hard to actually measure the time point of the cells. It's kind of a subtle point. So I can measure the profiles through the process, but I can't actually measure the exact time point that each happened at. And so this gave rise to this class of computational problems called pseudotime ordering, which says, can I take the cellular profiles without any of the time information and re-infer the time that each cell happened at, right? And it's a cool problem. It's a fun statistics problem, right? To assign each of these dimensional profiles to a time, right? And so, you know, we weren't the only people to think this. So this comes under this ordering part here. So there is like 12% of these 1,500 methods, or 1,100 methods rather, are for the pseudotime ordering idea, right? And you know, like 4 of these 129 are mine from my PhD, I think. So I was very much involved in this process. And I still think it's a very cool problem. The problem is, what's your baseline here? What is the baseline method for doing this, of fitting a single number to a high dimensional profile? And the answer is PCA, right? So there's this question arises of, well, is pseudotime just PCA, that we sort of realized more with horror as time went on. So if you think about PCA, it takes a high dimensional profile, whatever, and collapses it down to a reduced set of dimensions. And if I set that number to 1, then I'm getting a unidimensional number that could be time, right? And so we sort of realized this over the years, and eventually, so this is buried in this is our paper, buried in supplementary figure 11, which is where you don't want people to read stuff, where we started to have this question and just took a bunch of the common algorithms used at the time. I don't think people actually use it anymore. But you can run the algorithm and you can correlate the answer to this, to the first principal component of the data, right? And you see you're up sitting at like 0.9 for most of the data sets, maybe not this one so much. And you know, if you don't work with noisy biology data, like 0.9 correlation is like unheard of. That's like, that is really good. And the field sort of cottoned on to this time went on. So, you know, this is a more recent paper from 2020. And you know, they have this time series and they're trying to assign all these single cells to their times. You know, did they reach for 127 algorithms that can do pseudotime that people spent ages trying to come up with? No, they just PCA'd the data and use PC1 for their pseudotime, and you can't blame them, right? So I think the moral of the story here is, I don't think this is ever intentional, and there's certainly situations where PC1 isn't the real story, but a lot of the time it should be your first port of call. So I think it's a field where we're not very good at taking our baseline seriously, or indeed benchmarking when we're doing better than them. And all this sort of brings up to this idea that I started to be thinking about for a while, but it's put much more elegantly by Aki Vitari, who's a Bayesian statistician in Helsinki. So I've sort of stolen his graph, although I've redrawn it because his was much better than mine. But the point is, you can kind of think about this in a cost-benefit way, right? So if I think about this x-axis here, this is the effort I want to put into my statistical or data science model, right? So either the effort or what will end up being the model complexity, or some measure of how much time is being spent on this thing, right? And I can think about the y-axis as some notion of model performance, right? So maybe in a supervised setting, this is like some notion of accuracy. If I'm doing probabilistic modeling, maybe it's like the log marginal likelihood or something. But in either way, I can often calculate some metric of how well I'm doing. And I think the point is that if we're honest with ourselves, we know this isn't a linear relationship, right? I know that if I grab any machine learning model, if I spend twice as much time tuning it, I'm not going to get twice as much accuracy, right? So this leads to this super interesting question of like, when is good enough here? How far along this curve do I go before I'm like, this is good enough to hand off to the next level? And there's a whole bunch of other interesting questions here, right, as well. Like, so I've plotted two curves here. Like the pink is like, it's kind of worth going a bit further along it than it is on the blue. Do you know which curve you're on? Do you know how far along this curve you are? I think these are all fascinating questions that we need to think about more as a field to really do this. And beyond this, you know, is there like some, you know, I still love all the modeling. Is there some like meta model I could, you know, put on here that actually predicts for me whether it's worth keeping on going with the modeling I'm doing, or should I just stop and call it a day at that? So that's sort of, I think, an overview of where we're at with modeling in this field. So I'll just finish up with, you know, with some final thoughts about where we are as a field. You know, I don't mean to sound unenthusiastic about the field. It's an incredibly exciting time to be in it, and the data sets and types we're getting are really fantastic, and I think there's huge potential for discovery really throughout the life sciences with this. The caveats of this are really, we're often focused either on the wrong problem, or we solve the right problem over and over, or we don't realize it's solved. I think this keeps coming up again and again in our field that needs perhaps taken a little bit more seriously. The second is, you know, I'm not the first person to say this by a very long shot. We need much better integration with biologists and clinicians. If you're in machine learning, you call these people domain experts. This is hard. I think everybody knows it's hard. I still think it's 10 times harder than everybody thinks it is. We become quite siloed as communities sometimes with a lot of like domain specific jargon that makes it kind of hard to communicate over. So I think we need better approaches to this. I'm putting these out, by the way, absolutely no solutions proposed, by the way. This is really just a laundry list of issues. So if anyone has solutions, please email me. I think there's this incentive reward structure in academia that is interesting to think about, right? So what happens when the 80% solution works? What happens when I'm like a bit along this graph and my model's working really well and I haven't put that much effort into it, right? And the issue here really is if you're collaborating with a biologist, right, they probably just want a handoff and say, this is good enough, let's get going on the next part of the study, right? They're probably not going to want to wait for you for like 2 years to iron out all the tweaks in the model if it works well enough. You know, there's an education component to this as well. I've completely glossed over, right? That we need education of like what happens when you go really deep in a problem. You know, I was talking about this sort of rediscovering that PC1 is a really good estimator of this problem I spent in my graduate studies. Like, sure, it's mildly depressing, but it was a fantastic learning experience to go that far down the rabbit hole and really push with all these fancy models and learn all about this, right? So there's a huge educational component to this as well. Again, this problem, how do you know you're 80%? How do you know your model is working this well and can't be improved further? One person's 80% might be someone else's 20%. And you know, the final bit is, and this is again by far not an original thought, we still spend so much time manually locating, cleaning, organizing data. We spend a huge amount of time working through compatibility issues with various software libraries. There's this, I don't know if you've heard this phrase of graduate student descent, where you come up with a machine learning model and you basically tweak the performance by manually, essentially. And this happens all the time. And it happens in our field as well, where we're not, it's kind of interesting that I think in sort of pure machine learning, it's often you're just optimizing accuracies and likelihoods, etc. We do exactly the same sort of thing, but often on like benchmarks and heuristic measures. And you know, also like, could we have some methods that address these? And we're trying. We have some research sort of ideas in the group where we're actually trying to help automate some of these methods, but it's fiendishly hard. Like, I think there's a reason that it's not worked on, and the answer is it is hard. So anyway, that was a lot of thoughts of data science and life sciences. Hopefully it's useful. Thanks to all my group and our collaborators for all the discussions and leading many of these thoughts. And please get in touch. I haven't put my email address, but it's in places. Please get in touch if you want to chat or talk about anything else. And thanks for the invitation and the amazing seminar series as well.
Thank you so much for such a wonderful talk and for the biology 101 explanations, which I really appreciated. Definitely needed them. We'll just start off with the standard question, which is, what books would you recommend people read?
Oh, that is a good question. We still don't have very, I don't want to say this in case there is one I haven't seen. There's not much of the interdisciplinary between the ML and the biology. I think, like, for machine learning, something standard like Bishop or Murphy's would still be the best to dive into, like, practical introduction to the methods. For the biology, like, I think it's actually better to read a general interest book first to get you excited by the questions, and then once you sort of framed what's an important issue. There's a great book on cancer called The Emperor of All Maladies. I've actually forgotten the author's name, but it's like a history of cancer research for the last 100 years. And that's fascinating because it frames what the actual real important questions are that you can focus your thoughts around, if that makes sense.
Yeah, no, that's great. Thank you so much. I'll see if I can get a link to the book and throw it in the chat.